Rubina, tell me a little bit about what a blood culture test is.
That's a very, very good question. In the lab, we bring blood culture bottles in. Especially if a patient has a condition called sepsis. And sepsis is a bloodstream infection where a bacteria enter the bloodstream. So in the lab, we get this blood culture bottle, and our duty is to be able to identify a specific bacteria that is in the bloodstream, and then also be able to identify a particular medication that can be used to treat this patient.
Can you tell me how you would identify this bacteria?
It's a very, very interesting question as well. The first steps that we actually do is we receive these samples when they come to the lab. And once the samples are being received, we actually move on to put these samples in a very special incubators. And these incubators have a very specific temperature requirement. And if a patient is positive for a bacteria in the blood, within 18 hours to 48 hours, our protocol indicates that we go for about 5 days. The machine will alert us that this particular bottle is positive. And once it goes positive, the next step is that we have different kinds of plates that we actually use. And the purpose of this plate is that we're going to be able to culture this blood on these plates, and the plate has very specific media or nutrients in it, and it will be able to help us grow this bacteria. And once we actually plate this, we put them into another special incubator that also have a very specific temperature requirement, and it stays there for about 48 hours. And once that is done, it will go to the next stage.
So these plates help us identify what type of bacteria.
Excellent. These plates actually help us, and we have a very specialized trained medical lab technologist in the lab. And the next step is once these plates grow these bacteria, they will look at these plates and analyze it, and then they take the next step. And the next step actually involve identifying a specific bacteria in the plate. So we have a very special machine called the moldy. So this moldy is, we take a very small sample of this organism from the plate, and then we put it in this moldy machine, and then it will tell us whether it's an E. coli or it's a Staph aureus. So that's where the next step is.
And so if it is E. coli or Staph aureus or some other bacteria, what type of testing is done after that's identified?
So just to conclude everything, once you identify the organism or the bacteria, the next step is to be able to find a specific medication and a specific dose to treat the patient. So we also have another machine that we call a Phoenix. And this Phoenix, the purpose is to be able to identify a specific dose of medication to give to this patient. And so that's what this machine particularly do.
So really, in order for the patient to have a positive outcome to their stay, the doctors need that information about what type of bacteria and what type of medication to use.
That's the essence of the microbiology lab.